Hi, my name is Juliana Reves, and I will talk about this job I'm doing in the framework of my doctoral thesis. Well, we are studying how cells can act like radios by filtering temporal information, and in this framework, we are going to study how simple cell motifs can process temporal information by stimulating with square wave pulses, of which we can change the amplitude, the pulse duration, or the pulse period, and we are going to compare between the transient and regime response. That is, we are going to study how is the mean of the output between a time s and s plus t, where t is the input period, and with changing s value, we are going to change if we are studying the transient response or the regime response, where the transient response is the response of the system when it's stimulating and before it reached the regime state where the oscillation is periodical. We are expecting different kind of responses, like we plot here as examples. When we plot and we analyze the output in terms of the frequency for different systems. We are going to work with now with the Ligand receptor systems, and we are going to work with the dose conserves input protocols, where we are going to remain constant the total dose of the input. We are going to do that by changing input parameters between one period and the other to make that the color that represents the integral of the total dose constant. This is the differential equation for the ligand receptor systems. That is the system that represents the process where an external ligand binds to a cellular receptor, then they form a complex, and the complex desencadenates downstream responses. Our observable is Gs, that is the mean between S and S plus T, as we explained before, of the output, or that is the complex in the time. The input will be the ligand that is here in the differential equation. We found that for the two protocols that we consider, the mean value is different when we calculate in transient time compared with regime times. Here we can see that for low values of S, we have transient frequency preference that disappears for large values of S. That is because we call this phenomenon transient frequency preference. And we found this phenomenon for amplitude compensation protocol and duration compensation protocol. Then we studied how this comportment can be transmitted downstream when a covalent modification cycle is coupled to a ligand receptor system. Here, the ligand receptor is a kinase for the covalent modification cycle, that is the complex makes the substrates goes to be a product. We compare how is the covalent modification cycle transient frequency preference when upstreams we have different ligand receptors. Here we stay fixed the covalent modification cycle parameters in the same values, and we only change it how is the characteristical time of the ligand receptor by modifying the ligand receptor parameters. Here we compare how is a fast ligand receptor between a slow ligand receptor. We can see that in the slow case, there is a transient frequency preference that remains in the time during most times, but in both cases, it goes away for large values of S. And here we are comparing how is the response of the same node when we change the upstream node. We can see that in both cases, amplitude of the response changes between the time, but in the first case, where there is a fast ligand receptor response and the transient frequency preference is more lower than the slow case, the transient frequency preference of the covalent modification cycle also is very slow compared with the slow one case. That in this case we have transient frequency preference that is more robust and remains on the time for more time. As conclusions, we found that there is a transient frequency preference in very simple cell signaling models that can be transmitted downstream, as we analyzed before. We think that this phenomenon could be important and interesting when downstream nodes response is fast or the upstream stimulus is short-lived. That is, if a downstream node is fast, it's going to read this answer and not this answer. So that is important. Because of this, it's important to know how is the transient frequency preference, and also if the stimulus is short-lived, that is, here the stimulus dies, the important response will be the transient frequency preference. Thank you very much for listening.